Hi, I'm James Graham, CEO of Recce Pharmaceuticals Ltd, ASX code RCE. Thank you for joining today's webinar. Usual disclaimer. So we start today's webinar with a brief overview before our respective speakers join later in this presentation. For those who don't know Recce Pharmaceuticals, according to World Health Organization, we are clinically the most advanced new class of antibiotics in the world, and in fact, the first new class of antibiotics in over 40 years. We find ourselves in a privileged position with an advanced phase 3 trial underway at this moment for diabetic foot ulcer infections. We have multiple phase 1 and phase 2 candidates in unmet medical need patient populations, and we have a US Department of Defense program running in parallel. We have over 40 granted patents, and our products are on track to be commercially available later this year. As a snapshot of our company, to introduce ourselves, I actually started as the founding investor, supporting my grandfather, former head of Johnson & Johnson Research Australasia, so a third of the globe, in his retirement. Together with Michelle Delizia, we really made our first discovery to really identify a compound that works and keeps on working with repeated use, one of the greatest challenges of antibiotics or antibiotic resistance today. Various members, of course, join me in the board, and particularly you'll hear from today Dr. John Prendergast, our chairman, Michelle Delizia, our fellow board member and chief scientific officer, and Dr. Alan Dunton, our chief medical advisor and fellow board member. What makes Recce so different is we work broader than any other antibiotic known to date, and we work and keep on working with repeated use, which means we're valuable and useful today and will continue to be in the future. We work in minutes and not hours or even days like some traditional antibiotics, and we have multiple forms available or formulations available to target the bacterial species no matter where it is. I'm pleased to present our guest speakers today over this presentation, and we'll be joining you at the end of this presentation for a Q&A.
I'm Dr. Alan Dunton. I'm the Chief Medical Advisor, and I'm also a Director of Recce Pharmaceuticals Ltd. Today, I'd like to share with you some clinical highlights from our development program with our lead product, which is Recce 327. And actually, the lead product is the gel formulation. We also have an IV formulation, which has been studied. It's pointed to the excellent safety profile of Recce 327. We've given up to 4 grams of the product without any significant adverse events. We've never seen a serious adverse event with the intravenous product. So that's provided a really good background and understanding of the pharmacokinetics of the product for the other programs. We also have a spray, which will be used for respiratory infections, an inhalation product. We have some excellent preclinical data in mouse models of various very nasty bug infections like Acinetobacter baumannii, but that development is a little bit behind because we decided to focus on the gel product for diabetic foot infections. And why is that? If we could have the next slide, please. Infections of diabetic foot ulcers, next slide, please. It's a very complex environment. You can imagine that, thinking about your feet and your lower extremities. In diabetics, they have problems with their kidneys, they have problems with their vasculature. If any of you have members of your family that are diabetic and somewhat have had it for many years, you'll know that there's a lot of things that go wrong. They're also very susceptible to infections. They don't have good sensations in their feet, and they tend to bang their feet walking, even with shoes on, and those slight bumps can become infected. And it's very difficult to treat infections in these patients. And why is that? Well, they don't have good blood flow to their feet and their legs. So if you give an antibacterial or an antibiotic, by an oral route or an intravenous route, guess what? It may not even get there. They also tend to have a very high bacterial count, and there's very delayed healing. As I mentioned, they have problems with their immune system, so healing takes a long time. There's no standard of care of how to treat patients around the world, not just in any individual country. And most physicians who use these patients or take care of these patients, use cocktails of medications, and it's almost like an art rather than a science at all. Uh, when even you manage these local infections, as I mentioned, there's no standard of care. But the ideal would be where you could get an antibiotic directly on the wound. And that's why we are using a gel and why we're so excited about it, because our product has a novel mechanism of action. It's very fast acting. If you look in our test tubes and our in vitro studies against all bacteria, first of all, we've never had a bacteria tested that didn't succumb to the effects of Recce 327. And it happens very quickly. It's measured in minutes rather than in hours and to days, what most antibacterial products take to have an effect. And as I said, it has a broad spectrum of activity, and it's a perfect compound to use in this type of situation where you don't even know what bug you're treating or bacteria you're treating at the time. So you don't have to go around, take biopsies and see which bug is there. It's first line therapy that can be used right away. So we designed this Recce 327 gel with this complex situation in mind, because number one, it's easy to apply. You can imagine you open the tube and it can be applied directly. Um, you don't even need blood flow there because the drug is already there. And as I mentioned earlier, it has a very broad spectrum, and it could even work against an infection where you have many different types of bacteria that are present in the wound. So it's a perfect setup for the use of this kind of product. To my knowledge, there's no gel product approved, topical antibiotic product approved for use in diabetic foot infections. So if we can go to the next slide, let's talk a bit more about our product. There's the tube of Recce 327 gel. As I mentioned, you don't even have to identify the pathogen or the bacteria that's being treated. You can use it as an outpatient very easily. Um, and you don't have to expose the patient to a very high level systemically of an antibiotic. If you've ever been in antibiotics, they can cause havoc, especially oral products. They can give you upset stomachs, can produce diarrhea because it wipes out all the bacteria in the gut. Not a pleasant thing to have to take an antibiotic for 7 to 14 days. It's necessary sometimes, but we don't think you have to do it here. You can start with a topical gel early on in the course of the infection so it can be treated and not, get worse and have some dire consequences. We've thus done testing and I'll come to that later, the actual data, but you can see a response within 24 to 72 hours of the first dose of the drug. You can see a decrease in that redness and the swelling. And you can also see a decrease or you can stop the pus that's formed by these infections, which is basically white cells that the body is sending there to attack the infection. So it's a very rapid onset of effect. And when some physicians use oral or intravenous antibiotics, they can be on these things for weeks. We hope that's not the case in all of our testing, and we have some very good data, which I'll share later in the presentation. It's very safe. We've looked at how much drug is absorbed from intact skin where the drug is applied, or even in these wounds. If you have intact skin, there's absolutely no drug that gets into the systemic circulation. And in a diabetic foot ulcer or other types of infections where there's an open wound, you can see very, very small concentrations, which it amounts to about 50 milligrams given systemically, which is a very, very small dose of this drug. So it's a very, very low. And even if it was absorbed, the drug is so safe, as we showed in the intravenous program that we have, there's no ill effects that it causes upon the body. And I've used it on a few infections I've had in my hands and my feet, And it's a clear gel. It's very soothing. It actually feels pretty good. And you can use it once daily with a small bandage on top that's put in place. Next slide, please. So let's move on to this program. It's an interesting history, actually. So the first study we actually did was with a liquid spray formulation of Recce 327. And we studied patients who had diabetic foot infections. It was a hospital it's called Liverpool Hospital in southwestern Sydney Health District. And we studied a handful of patients out there, and lo and behold, all of them responded to the drug. And in fact, in some of them, over a very short period of time, we were seeing these diabetic wounds actually starting to close and to heal completely, which is an incredible feat. Uh, Pardon the pun, but most of these wounds take a very long time to heal. Now, one thing, you need to heal the infection before this open ulcer has any chance of closing. So without this treatment, the patient wouldn't have had a chance to heal their wounds and close it and prevent any further reinfection of the wound. So that we looked at that data and say, well, let's go after a gel formulation, which would be much easier to use. We also did a phase 1 study in patients who had infected burn wounds, and we showed that it was safe. I believe none of the patients had any serious adverse events. One patient had a little bit of tingling. Patients with burns have very sensitive skin. Their nerves have all been shattered, if you will. And there were no serious adverse events in this study. It was done at a hospital in Western Australia near Perth, which specializes in burn treatment. And we were very pleased with that. So by this point, we had a gel which we were going to put in clinical trials, but we also used another way to understand the drug a little bit better. The Therapeutic Goods Administration in Australia has something called a special access scheme, where any physician can request an investigational product um, to treat their patients under their care, as long as they abide by the TGA's requirements, which is basically to ask permission, and if the company says yes. Well, we said yes to a number of these special access scheme requests, and we saw incredible results. And I think one of the physicians who's an orthopedist in North Sydney used it on a number of patients, and one patient who had an infected ulcer on his ankle. Uh, he saw a response within 24 hours. He was amazed, and that patient went on to completely heal that ulcer in very short order. And he had a number of other very difficult patients that he couldn't cure with oral antibiotics, and he used the gel, and it resulted in a cure. So he's been very pleased with the product and is looking forward to further developments as well. We did proceed to start a phase 2 trial and complete a phase 2 trial, at least the first part of that trial, in patients that had something called an acute bacterial skin and soft tissue structure infections, affectionately known as ABSSSI. I'll come to a little more extended data on that, but the the high percentage of those patients, I think it was greater than 90%, achieved a positive primary and secondary endpoint. It was healing of the ulcer from the infection, and the patients did very well. It was safe. We looked at absorption of the drug, as I mentioned earlier, and it was minimal absorption. Um, this study also included patients with diabetic foot infections. They're in the broad category of ABSSSI, and they also responded nicely as well. So we decided to focus on diabetic foot infections going forward, and I'll talk about a little bit more about that in a couple of minutes. But if I can have the next slide, please. So this is the more complete results of that phase 2 study in patients with infections on their skin or in their skin structure infections. Um, It enrolled 30 patients. There were 29 included in the final analysis. There was a patient that was withdrawn. She had some pain which existed before She entered the site and the pain became problematic for her, so she dropped out within the first or second day of the study. But other patients went on to complete the study, as I mentioned, these 29 patients. And after 7 days of treatment, 7 days of once daily treatment, 86 of the patients had a positive clinical response, a successful clinical response. And we continued treatment for 14 days, and that number increased to 93% of patients. That's a really astounding number in a phase 2 trial. This was an open label study, meaning there was no comparator in the trial, there was no placebo or active other antibiotic that used in another set of patients, but still was dramatically positive to the point that we felt we could move into a phase 3 study. It was very safe. There was no adverse events or serious adverse events. And I should just point out that the assessment method is something called the Lipsky scale. For the infection, you look at 7 parameters such as redness and swelling and exudate and pus exudate, etc. And they're scored from 0 to 3, three being the worst, if you will. It's really bad. For 7 parameters, you can have a score as high as 21. So a positive in this study was a significant improvement or an absolute cure over that course of therapy. It takes time for the body to respond. You can kill the bacteria in minutes, but the body still has to catch up with the response and you slow down the immune system, if you will, to get rid of the redness, swelling, and in any cases, irritation as well. So we were so pleased with that study, we proceeded to another study on the next slide. If I could have the next slide, please, which is a phase 3 trial. We were actually working with the government of Australia and speaking to folks in Indonesia because they have a huge problem with diabetes in Indonesia. And they have so many patients that have infected feet. Infected diabetic foot ulcer infections because that environment is very difficult, right? It's very humid. Um, they wear primarily sandals or flip-flops, not complete shoes. Their diabetes, unfortunately, is not treated aggressively. So we began at the end of last year, 2025, a phase 3 trial, which was approved by their local regulatory body in Indonesia called the Biden POM, BPOM, BPOM. And that study has been running now. It's where are we today? Is the middle of March. And we're open at 5 sites across Indonesia, from the west to the east. As you can see, you need to have the population of Indonesia is very diverse, subpopulations, et cetera. So the BPOM wanted us to study across the geography of Indonesia. The primary endpoint here is the clinical response to diabetic foot infection, according again to the Lipsky scale. And the secondary endpoint is the total wound score, which is a measure of how wide and how deep the ulcer is primarily, and also obviously the safety of 327 gel. So really, really excited. We'll be performing an interim analysis this year when we complete 155 patients. We're going to enroll approximately 300 patients total, and then we'll look forward to registering the drug first in Indonesia. But the good news too is Indonesia is one of the ASEAN countries, and if you get a drug approved in one of the ASEAN countries, it opens up the door to all the other ASEAN countries as well to seek approval. So we're very excited about this evolution. And as I mentioned when I started the talk today, we're working in other areas. We have an IV formulation. We also have an aerosol formulation for lung infections. It's becoming very popular now to try to treat patients with an inhaled version of an antibiotic for the same reason we're giving a topical gel. You're getting the drug to the site of action where it is most needed, and you're not exposing the rest of the body to that drug. I think that's the last slide, if we can move this one forward. But I think it is, and I really want to thank you for your attention, and keep your eye out for more good news from Recce Pharmaceuticals. And have a good day. Thank you so much.
Good day, everybody. My name is Nathan. I'm the CEO of Etana, and today I would like to introduce about Etana and Recce Corporation in Indonesia. Next. First, I would like to introduce about Etana. So Etana is one of the few biopharmaceutical company in Indonesia. Indonesia, there's only three vaccine company. We're one of them, and there's only two company focusing in biotherapeutic, and we're one of the two. So as a company, we're really focusing in the whole biopharmaceutical field. And one of the key purpose for us is really to provide a better standard care for the people in Indonesia. And that's why we're very excited that today we have a very strong collaboration with Recce Pharmaceutical to bring their new product, their new technologies to the Indonesian patient in Indonesia. Next. So with partnership with Recce, we also go through a very strong collaboration by doing a late-stage clinical trial in Indonesia. Uh, this will allow the Indonesian patient to get first in hand the treatment. It also gonna help tremendously the clinician how to treat the patient better with Recce technology. We also get a full support from the both government, both from Indonesian government, both from Australian government to perform the clinical trial. It actually has been quite a very long time that there has been a very strong collaboration between two pharmaceutical companies in Indonesia and Australia. So this has also marked a very significant milestone between the cooperation between two countries. So we really hope that the clinical trial of Wiki will land very successful because it will not just also mark the collaboration successful, but also mark in terms of uh, the impact that we can do for the Indonesian patient. Obviously, what we can offer bring us, a tremendous opportunity um, to have a better standard care in the Indonesian healthcare, which is actually really, really need a lot of upgrade. Next. So with Ricky, we're doing phase three clinical trial in Indonesia. We're gonna involve hundreds of clinical trial patients, and then we hope we can complete it within this year. And we can also hope that with the trial completed this year, we will also gonna get the approval this year. So the topical product, although looks simple, will present a very major step for our cooperation in treating diabetes food for patients, where in Indonesia we don't really have a lot of option. And by Ricky providing that technology, will provide a better option to support a patient need. So we really thank you for the collaboration between us and Ricky, and we're really sure that this product could really contribute um, to the better safety and better health for the nation patient. Thank you so much.
Hi everyone, my name is Dr. Jane. I'm currently stationed as the Clinical Research Department Head in Siloam Hospitals, Siloam Clinical Research. A bit background for Siloam Hospitals. We are currently the largest private hospital network in Indonesia, and we are aiming to become the trusted destination for holistic and world-class healthcare. Having 41 hospitals, 76 clinics across 28 cities in Indonesia, we are in the position to advance technologies in healthcare, and we are always inspired to provide the healthcare that we can have for our Indonesian patients. We've been in these industries for more than 30 years now, and we are in the position to have so many firsts in Indonesia. So we are the first hospital in Indonesia to have the first GCI accredited hospital. We have our top ranked private center hospital as well in MRCC in Jakarta, and so on and so forth. And one thing that we realized especially during the COVID pandemic, is that if we want to keep advancing in technologies in healthcare, we need to also invest in clinical research because it is the future of medicine. Cannot just deliver services without contributing to the research welfare as well. So that is why 5 years ago we decided to build this department in the hopes of being the one-stop center for all the clinical research activities in Siloam Hospitals. Because we do believe that clinical research is the foundation of advancing healthcare. It will ensure that the medical intervention are not only safe and effective, but also relevant to the people that we serve. In Indonesia alone, much of our medical practice has historically relied on research from other countries. While these global studies have offered great insights, such findings may not always address the specific health needs for our local populations. In Siloam, we have managed to bridge this gap, and it is a clear mission for us to contribute meaningfully to global research while we strengthen the quality and relevance of clinical trials for Indonesian patients. So far, our portfolio has rose to 32 projects in the last 4 years. The top therapeutic areas would be oncology, infectious disease, and neurology. We've been receiving many tropical diseases trials. I would say that it is contributing to the factor that we are in a tropical country. And that leads us to one of our exciting projects with Recce Pharmaceuticals. We've been introduced to this project around 2 years ago, and we know that through these clinical trials, the work with Recce Pharmaceuticals, we are able to provide some access to our Indonesian patient who has a diabetic foot ulcer condition. And it's one of the way to find a new treatment regimens to the current DFU populations. We are looking forward to continuously support Recce Pharmaceuticals and this clinical trial. We have been working with two Siloam hospitals, one in Siloam Hospitals Lipo Village in Karawaci, and then the other one is in Bali, Siloam Hospitals Denpasar. And we've supported them with the patient recruitment, doing the informed consent, monitoring the visit. We do the follow-ups, and so far, all the patient has been treated with this either Recce or the placebo that we provided. All in all, we're very excited. We will look into the preliminary result later on, and I'm looking forward to finish this study. Thank you so much.
I'm David Lassiter, the founder of and partner at Horizons Global Solutions, and we've been working with Recce now for a couple of years to develop the strategy and to execute this strategy. And working with the United States federal government and specifically right now with the Department of Defense or Department of War. My previous experiences, most recently with the United States government, were serving as the Deputy Assistant Secretary of Defense for countering weapons of mass destruction. And in that role, I oversaw all programs, policies, and strategies related to seaborne defense, both internationally and working with U.S. government interagency partners. And one of the major issues that I oversee was the biological threat response and spectrum. And what Recce 327 is doing is exactly what the Department of Defense, Department of War are seeking. Ryan?
Thanks, David. And Ryan Kane, Managing Director of RK Strategies. RK Strategies is a healthcare, life sciences, and preparedness lobbying consulting firm helping biodefense companies, public health companies navigate the U.S. government. Simultaneously, I also serve as the Vice President of a leading biodefense company and previously worked for several United States senators, members of Congress, mostly in political, but also policy capacities, and eventually turned to the private sector. David and I have been working together very closely to help develop Recce's U.S. government programs, and we're excited to share more with you today about that. Next slide,
Next
please.
slide. And as we've been working together, most of it is focused on ensuring that what Recce is doing is answering the call of the United States government, and in this case, for the Department of Defense and the warfighter. And when we talk about providing the best capabilities, we also talk about doing this at the point of care, doing this on the battlefield to provide a capability to as quickly as possible to the warfighter, that being either, again, on the battlefield or at the closest level of care. This is both for antimicrobials, but also, and probably most importantly, in a lot of the research that Recce is undertaking now with the United States government, that is in burn and blast wounds as well as combat wounds. And so this is a very important area that the department wants to continue to try to get a hold on. Ryan, other thoughts?
Thanks, David. Other areas the government's looking very closely at, of course, is antimicrobials, which continues to pose a significant challenge. We've seen that out in the field, for instance, in Ukraine, Israel, and other areas in the theater where there is significant infection on the battlefield, and current antimicrobial candidates are not able to support those wounds. And so all that to say, the U.S. government has looked very closely at R327 and has supported it through several programs, in large part because of its broad utility as a new antimicrobial candidate that also addresses the burn and blast areas, combat wound infections, and of course, biodefense threats. I would also point out that the U.S. government has moved towards the adoption of pathogen agnostic approaches. Think of drugs that can treat multiple bugs versus the old model, which was one drug to treat one bug. And that model's failed in the U.S. government. And so they've moved to look at rapid treatment options that can treat a wide array of threats, including threats that are unknown, that are easy to scale and shelf stable, and this sort of thing. And R327 really fits the bill nicely. Next slide, please.
And as we discussed a little bit previously, the challenges for the United States military are quite significant in this area. We want to be able to save the limbs, to stave off infection, to provide a countermeasure as quickly as possible, because it will the department would then have a better chance of, again, providing the best solution to those wounds, and then preventing the potential loss of limb, and even more than that, life. And so most of these are multiple potent bacteria. So something like R327 has the ability, as we've seen, to kill all of it. And that's absolutely essential because it enables that application to be applied to any wound in any climate and place, which is something the Department of War obviously seeks routinely, as a global environment for potential operations. Included this would be, obviously relief for pain that the warfighter might experience, and to promote essential wound healing. And I know, Ryan, we talk about antimicrobial resistance being, only growing and growing as the years go out, and such a capability as 327 will provide the right solution for the United States government, and in this case, we've discussed the U.S. warfighter and our allies. Over to you.
Thanks, David. And the other area I would add is that there's broad application as well within the biological threats. These are category A and B agents, agents that are incredibly deadly and that are significant concerns. Adversaries to the United States and to our allies are actively developing some of these agents today, and the U.S. government is looking to develop medical countermeasures that may treat it. And R327 really fits the bill nicely. And as we'll talk about in a future slide, the compound's been tested and is in testing against a number of these category A and B agents. Meanwhile, it's also highly efficacious against more of the common infections. And so across a variety of formats, a gel, a hydrogel, a number of other formats as well, such as IV, where there's a lot of different routes of administration to treat a lot of different infections, and that really drives the utility of this compound. Next slide.
Yeah, and we're really excited. We've been working on three areas in particular with the U.S. Department of War. The first is the congressionally directed medical research program, and it's an annual program that the United States Congress funds specific areas for the Department of Defense, Department of War, to research and provide capabilities, medical countermeasures, and other research to the department. And it's a really important program. In this particular opportunity that Recce was selected for, as providing a synthetic anti-infective, as we've talked about, R327, for treatment of burn wounds and the downstream impact. And this has allowed for the work and development of this room temperature stable 327 hydrogel dressing in sachets for field use. And this will allow the treatment on burn wound infections, and the current work is on animal models, and it's showing some success. And it's been great to see thus far, and I know the department is eager to continue this work with Recce. Ryan?
And that award, the CDMRP, is a highly competitive award. In fact, it's one of your most competitive grants that a company can receive. And so it's especially notable that Recce, an Australian company, was able to win this award. To win this award involves a very significant amount of vetting at the highest levels of thought leadership in the infectious disease space. And Recce was able to come out on the top and secure that. And that was a several $million contract that supported this program and continues to this day. Additionally, there are two other programs that are underway, both CRADAs, which is cooperative research agreements, research and development agreements. The first one with the U.S. Army, U.S. AMRID, which is the U.S. Army Infectious Disease Institute, and that's looking at a number of the category A and B indications that I mentioned on the prior slide: Burkholderia, Yersinia pestis, Pseudomonas, this sort of thing. Some of the worst pathogens that are causing the most problems or of the most concern to the U.S. government. And this testing is well underway today in a U.S. government laboratory. And that program is funded today through the Defense Threat Reduction Agency support into that U.S. AMRID lab. The other program is at the U.S. Army Institute of Surgical Research, looking at reducing the bioburden of Pseudomonas and burn wounds. And that's yet another extension of the ongoing U.S. government efforts. And all this to say, with the two CRADAs and then, of course, the grant, it just underscores the promise of R327 in the eyes of the U.S. government. They're really excited about it because it's fitting the need of the U.S. government and continue to invest in it. And we would expect to see more in the future, given the wonderful capability that this compound brings. Over to you, David.
Yeah, I think you nailed it there, Ryan, at the end, that these three opportunities are, in our experience and our knowledge, are likely to lead to other bigger and even better opportunities with the United States government, which we're excited about. And there are even a couple of opportunities this year that we're already surveying, and we believe there's a high likelihood of success that Recce 327 could be very competitive to winning some of these even larger awards. And so we're looking forward to that work. We know this is a many-year effort, and it's going to pan out to be quite incredibly capable for the United States warfighter, and as we talked about, our partners and allies as well. Thank you.
Thank you all.
Hello and welcome. My name is Michelle Delizia, and I am Recce's Chief Scientific Officer and an Executive Director. I work as a medical scientist and medical microbiologist, and my background with Recce began at its inception some 13 years ago when I joined with its founder and inventor, Dr. Graham Melrose, ex-Johnson & Johnson, and a pioneering polymer chemist. Together, we worked to develop our lead candidate antibiotic compound, Recce 327, and establish a recognised and globally patented brand new class of antibiotic technology to tackle the increasing threat of antibiotic resistance to worldwide healthcare. In this presentation, we will take a look at some of the contributors to antibiotic resistance and some of the areas of very high unmet medical need where antibiotic resistance is increasing and infections are getting more and more difficult to treat or even not able to be treated with our current and traditional classes of antibiotics. I will present some compelling preclinical data that demonstrates the consistent and powerful efficacy of R327 to treat serious infections, particularly those characterised by antibiotic resistance, such as pneumonias in hospitals and in the ICU, and tuberculosis. Finally, we will consider how this solid preclinical data establishes the foundation for new and successful treatments in the clinic and in the face of dangerous drug-resistant bacterial infections. So there are multiple drivers of antibiotic resistance, all of which have contributed to serious infections and which continue to threaten and weaken the ability to treat common infections. These infections then go on to become serious and life-threatening. For example, and looking at the various points on this slide, there is an overall and widespread overuse and inappropriate use of antibiotics in both animal care and human health care. This mass exposure has given bacteria unprecedented opportunity to adapt and mutate against these antibiotics, developing resistance to the point where we now have antibiotic resistance in every class of our current and traditional antibiotics. Taking a look now at VAP, which is ventilator-assisted pneumonia, multi-drug-resistant pathogens are a primary feature of this serious pneumonia. VAP is considered a subset of HAP, which is hospital-acquired pneumonia. And VAP typically occurs after about 48 hours of mechanical ventilation. And this is when microaspirations of pathogenic bacteria in the throat are inhaled. They travel down the endotracheal tube and straight into the lung tissue, which is highly vascularized and so vulnerable to infection. Now, this significantly increases the risk of patient death, and mortality rates are very high in these patients, ranging from 30 to 70%. Common pathogens in this scenario are the notorious and increasingly drug-resistant gram-negative bacteria such as Pseudomonas and Acinetobacter. So looking also further at VAP, we've mentioned that the prevalence of multi-drug resistant pathogens is a big driver. And so, though, is the increasing weakness of our present arsenal of antibiotics. For example, most patients, 60%, do not respond to first-line therapy. And this dire situation only increases their time on mechanical ventilation and all its risks, and leaves just further time for the critically ill patient to deteriorate. However, we will see in the following slides that Recce 327, as a brand new class of antibiotic, effectively treats multi-drug resistant pathogens such as we've seen in VAP and HAP. So, for example, looking at this pivotal proof of concept data we have in a validated mouse model of HAP caused by carbapenem-resistant Acinetobacter baumannii, let's call it CRAB. Now, this organism has been identified and tagged by the World Health Organization in its priority pathogens list. And it's been allocated the most serious category of critical significance in view of its resistance to last resort antibiotics, severity of the infection it causes, and the significance of its global health burden. In this model here, Recce was successfully nebulized for delivery straight into the infected lung tissue, enabling direct treatment at the various lung sites of infection. After only 24 hours treatment, the bacterial burden in the lungs was reduced by a remarkable 4 logs, which 99.99% of bacteria, and very close to the limit of detection in this assay. We've also administered Recce 327 intranasally, and both intranasal and nebulizer resulted in significant bacterial reduction in the lungs, and it was also well tolerated. It's noted that the comparator antibiotic was meropenem, which is not able to be nebulized, hence it does not have that direct lung delivery that's possible with Recce, and it's also not well tolerated with known side effects in the gastrointestinal tract. A second and highly significant global infection is tuberculosis, and this one is also in the WHO priority pathogen critical group. TB is considered one of the world's top infectious disease killers, taking out around 1.5 million people every year and around 10.8 million new cases every year worldwide. It's a particular burden in Indonesia, where Recce is currently conducting a major clinical trial to treat diabetic foot infection. Indonesia accounts for about 10% of global TB cases, and making it one of the largest TB healthcare markets in the world. And we'll see in the following slide how Recce 327 is uniquely and strongly placed to address this market of very great unmet need. We have here on this side good and solid preclinical data in a validated model of TB infection in mice using the related organism Mycobacterium fortuitin. It's a recognised proof of concept model. And after 7 days of intravenous treatment with Recce 327, the mice exhibited significant decreases in bacterial load in their kidneys compared to the vehicle controls. So it was a bacterial or bactericidal 3-log reduction, well tolerated, correlated with reduced kidney lesions. It was a result that was superior to the comparator antibiotic rifampicin. So we've seen, of course, very good preclinical data. The question then, of course, is, okay, how is this going to translate in the clinic? And pleasingly, we have moved from standard and reference validated in vivo models. We also back that up with testing clinical isolates of bacteria from the Centres for Disease Control from their infectious disease bank. And we tested Recce along with common everyday antibiotics against nearly 700 clinical multi-drug resistant isolates. And in all cases, they were susceptible to Recce. However, in the comparator antibiotics, a significant proportion were resistant. Coming to our conclusion now, another significant and very pleasing feature of Recce is that it acts very quickly, within minutes and not within hours. So we're looking at a much less exposure time to affect potential systemic toxicity. In this assay, we tested against several common everyday antibiotics, all of which have tried to tackle bacteria in various ways through mechanisms of action. For example, bacterial cell wall synthesis, that's cephalexin, DNA replication, that's ciprofloxacin, cell membrane, colostin, and protein synthesis, the tetracycline. And Recce outperformed all of these classes of antibiotics, and it did so quickly. It's been my pleasure to present this bank of outstanding preclinical data to you. I believe it resoundingly demonstrates the unique and powerful properties of the Recce 237 antibiotic technology and its ability to tackle the rising global threat of antibiotic resistance. Thank you.
Hello, I'm John Prendergast. I'm the Executive Chairman of Recce Pharmaceuticals, and I'm delighted to be with you today to present a company overview and to give you a flavour of what we anticipate over the next 12 to 24 months. But before I do that, I want to draw your attention to the fact that there are almost 5 million deaths globally from antibiotic-resistant infections. Roughly 75% of antibiotics in development are derived from existing drugs, and it's inevitable that bacteria will develop resistance to these newly derived antibiotics. So what does that mean? The antibacterial pipeline is in crisis, and as the WHO has pointed out recently, last year, this is due to a scarcity of products and a lack of innovation. So much so that in the past 10 years, only 2 have represented new chemical classes. Effectively, this represents that the development of innovation in antibiotic space is essentially stagnant. So when it comes to our company, we are looking at being truly innovative, just like other disease areas that have had high-packed innovative products over the last 20 years. For example, in the arena of cancer, PD-1 antibodies have transformed the treatment of certain cancers. And in the area of obesity, which has been such a growing global problem, GLP-1 inhibitors have impacted that dramatically. But when it comes to infectious diseases, there are no truly new innovative products. Recce's polymer platform represents the opportunity to really innovate because it's a fully synthetic polymer platform that's designed to disrupt the essential component of any growing cell, particularly bacteria, and that's the production of energy. The World Health Organization recognized it last year as the only molecule that stands out as a fully synthetic compound that targets ATP production, which is the essential energy production arena within a bacteria or any growing cell. We've gained from knowing that we have a truly innovative product has allowed us to develop a program pipeline depicted in this slide. We have wanted to access the market as quickly as possible with this particular product, and consequently, we've developed a program in diabetic foot ulcer infections that originally was conducted in Australia, but because of the enormous opportunity in diabetic foot ulcer infections in the ASEAN region, particularly in Indonesia, where there is a population of 280 million people, with 10% of the population having diabetes, this has allowed us to develop a registrational strategy where we are conducting a phase 3 trial in Indonesia. And with that registration in the course of the next 12 months, that would allow us to expand us into the ASEAN region. Behind that, we will continue to conduct our topical gel program in Australia that would then allow us to move over to other additional registration clinical trials that would support a registration package for the US. Underpinning this all is a safety program where we administered Recce 327 IV in over 150 human volunteers, where we saw no aberrant safety parameter or physical effect of the drug in any way, shape, or form. In addition to our lead program, we're looking to expand our pipeline through the inhalation of Recce 327, particularly applied to lung infections. And these particular lung infections are hospital-acquired pneumonia and ventilator-assisted pneumonia. We have already conducted studies to demonstrate the efficacy of Recce 327 when it's delivered to the lung. And so these two programs this program would also allow us to not only access those types of infections, but potentially be applicable to other diseases such as tuberculosis and potentially cystic fibrosis. This truly represents an exciting program. We have recently been awarded a $2 million grant by the US Department of War that would further our opportunities in the topical gel space, particularly as it is applied to the war combat veteran, where in the field, any particular type of wound could be treated with a topical gel, as well as burn wound infections that we are conducting under a cooperative research and development agreement with two other agencies in the US Department of Defense. As we roll out our commercial strategy, as I mentioned, we are conducting the phase 3 trial in Indonesia, and that allows us to access the other Asian countries by virtue of the harmonization guidelines between regulatory jurisdictions. Moreover, that opportunity would also lend itself to expanding our reach into the Middle East, North Africa region. Why so? Because between the two regions, MENA and ASEAN, there are over 120 million diabetic patients, of which there would be a representation of approximately 10 million diabetic foot ulcer infections. So this is a significant market. In conjunction with that, we'll continue our regulatory and manufacturing base in Australia, and those critical studies will then allow us to expand into the world's largest infectious disease therapeutics market, which is obviously the USA. So we're very confident of being able to adopt an impactful global commercial strategy. So when we look at Recce today, I want to present several key takeaways that really underpin the future value of the company as a true innovator in the infectious disease space. And it's important to recognize that this particular polymeric platform is broad spectrum because part of the significant problems associated with traditional antibiotic development has been the lack of antibiotics to target more than one bacterium at any one time. We target bacteria independent of resistance to other antibiotics. More importantly, we've tested the polymer in over 750 clinical isolates and never failed to kill the bacteria within minutes. That's part of the effective mechanism of action. It's not in hours or days like traditional antibiotics, but it demonstrates amazing lethality in minutes. And in passaging experiments, which are truly the standard bearer for determining whether an antibiotic has the ability to kill multiple times, we've tested in passaging series of up to over 100 times and never seen development of resistance. As I mentioned before, in our clinical studies, one of the key things going forward with these programs is to show that the product is safe, and our safety profile is outstanding. When we consider what we are trying to achieve with the topical gel, we want it to be targeting the site and acting rapidly at that site, unlike oral or IV antibiotics, which can take hours to days to be effective. As we progress our studies of topical gel and success with our phase 3 program, we believe that this will become the standard of care for diabetic foot infections globally. And so would afford us the opportunity to price the product in a premium manner. In fact, all our pipeline opportunities are driven towards premium price therapeutic areas and will be used on a global commercial basis. Currently, we've had several reports that have valued the company significantly above where our current market cap is. Whilst this is really reassuring and represents opportunity, what I truly want to reflect is that the innovation and the other pipeline opportunities that are available within the company over the coming years have not been necessarily reflected in these valuations. So we believe that the company has true upside above and beyond these current projected valuations. And why so? Because Recce is a pioneer in the development of a new era of anti-infectives. It has the world's first synthetic anti-infective platform that we believe will be able to engineer on an ongoing basis to outsmart superbugs and the superbug crisis. It is the first novel class of antibiotics in over 40 years, and we are rapidly de-risking this through our registrational phase 3 trials in Indonesia and potentially in Australia. This will become the first topical anti-infective approved for the treatment of DFIs, and more importantly, from a global perspective, will be the first standard of care. We have multiple value-creating opportunities across a series of other pipeline programs that meet unmet global needs. As I mentioned, particularly in the area of lung infections for hospital-acquired pneumonia and ventilator-assisted pneumonia. All these programs are designed to impact multi-billion-dollar markets that are in dire need of innovative treatments. So what I'd like to leave you with is that Recce is a pioneer, certainly, but we want to become a global leader in the anti-infective space, and we truly represent the first broad-spectrum effective antibiotic/anti-infective in over 40 years. I want to thank you for your attention, and I look forward to updating you over the course of the next 12 months on our programs. Thank you.